Hello guys, welcome to our channel AskPrep. Today's series is for 11th class biology. We will be studying Chapter 8, Cell the Unit of Life in detail. We have also added important notes for your exam preparation. All the best for your exam. Let's begin with the discovery of cell. The discovery of the cell was facilitated by the invention of the microscope. In 1665, Roberto improved the compound microscope. Hooke observed cork under the microscope and saw small structures resembling chambers, which he named cells. Antony van Leeuwenhoek, a Dutch scientist, perfected a simple microscope with a single lens. Leeuwenhoek's microscope could magnify objects 200 to 300 times. Leeuwenhoek discovered microorganisms like bacteria and protozoa. These breakthroughs led to the exploration of the microscopic world. Next topic is cell theory. It consists of three core principles. The first part, proposed by Theodor Schwann in 1839, states that all organisms are made of cells. Schwann and Matthias Schladen expanded this by declaring that cells are the basic, structural, and organizational units of organisms. Rudolf Virchow contributed the third principle in 1858, which asserts that all cells come from pre-existing cells. These principles form the foundation of cellular biology and understanding of living organisms. Overview of cells are classified into two categories: prokaryotic and eukaryotic. Prokaryotes include unicellular organisms from the kingdoms bacteria and archaea. Eukaryotes include animal cells, plant cells, fungi, and protists. All cells share four common components. Plasma membrane acts as an outer covering separating the cell's interior from its environment. Cytoplasm, jelly-like region where various cellular components are located. DNA, the genetic material responsible for cell function and heredity. Ribosomes, particles essential for synthesizing proteins. Prokaryotes and eukaryotes differ in internal complexity and the presence or absence of a nucleus. Prokaryotic cell. Prokaryotic cells are single-celled organisms without a true nucleus or membrane-bound organelles. They belong to the domains bacteria and archaea. Prokaryotic cells are enclosed by a cell membrane, providing protection and maintaining shape and moisture cells. These cells contain free-floating DNA and ribosomes. Prokaryotic ribosomes are not attached to the membrane. Eukaryotic cell. Eukaryotic cells have a clearly defined nucleus enclosed within a nuclear membrane. The nucleus contains chromosomes. Eukaryotic cells possess various organelles like mitochondria, Golgi apparatus, endoplasmic reticulum, and lysosomes, each with specific functions. Eukaryotic cells are larger and more complex than prokaryotic cells found in bacteria and archaea. Difference between prokaryotic cell and eukaryotic cell. See the image. Next is cell structure and function. Here's an overview of some key cellular structures and their functions. Cell membrane, plasma membrane, acts as a barrier between the cell's internal and external environments. Regulates the passage of substances in and out of the cell. Ensures essential molecules enter and waste products exit the cell. Cell wall in plant cells provides structural support and protection. Maintains cell shape and prevents bursting from internal pressure. Allows the exchange of water, nutrients, and gases between adjacent cells. Cytoplasm, gel-like substance filling the cell's interior. Houses organelles and supports chemical reactions. Facilitates the movement of organelles and cellular components. Nucleus contains the cell's DNA and controls activities via gene regulation, surrounded by the nuclear envelope, separating it from the cytoplasm. DNA is organized into chromosomes within the nucleus. Organelles. Organelles are specialized structures within the cell that perform specific functions. Examples include mitochondria, responsible for generating energy in the form of ATP through cellular respiration. Endoplasmic reticulum, ER, involved in protein synthesis and lipid metabolism. Rough ER has ribosomes attached to its surface, while smooth ER does not. Golgi apparatus modifies, sorts, and packages proteins and lipids for transport, within or outside the cell. Lysosomes contain enzymes that break down waste materials and cellular debris. Chloroplasts in plant cells, site of photosynthesis, where light energy is converted into chemical energy in the form of glucose. Fluid mosaic model proposed by Singer and Nicholson, 1972. Describe the cell membrane structure as dynamic and fluid. Lipids form a bilayer with proteins embedded, creating a mosaic pattern. Lipid molecules are fluid, allowing lateral movement of proteins. The fluid nature contributes to the membrane's flexibility and adaptability. Functions of cell membrane. The cell membrane, composed of 52% proteins and 40% lipids, serves several crucial functions. Functions of cell. Membrane, physical barrier, separates the internal environment from the external, protecting cellular components and enabling distinct internal and external activities. Structural support, maintains cell structure by supporting the cytoskeleton, a network of protein fibers. Selective permeability, controls the movement of molecules in and out of the cell, allowing essential substances, e.g., water, oxygen, carbon dioxide, to pass through while regulating ion and polar molecule movement. Endocytosis and exocytosis facilitates endocytosis, engulfing large molecules, particles, and exocytosis, releasing substances into the extracellular space. Cell signaling. Specialized proteins and carbohydrates on the membrane surface play key roles in cell signaling and intercellular communication. Next is cell wall. Structure. A rigid layer surrounding the plasma membrane found in plants, fungi, bacteria, algae, and some archaea, not in animal cells. Plants, composed primarily of cellulose fibers, glucose, and polymers. Bacteria, composed of sugars and peptidoglycan. Fungi, contains chitin, glucans, and proteins. Layers in plant cells, middle lamella, rich in pectins, facilitating cell adhesion. Primary cell wall, contains hemicellulose and pectin, found in developing plant cells. Secondary cell wall, reinforced with lignin for strength and water conduction in some plant cells. Functions. Provides structural support and regulates cell growth. Controls molecule diffusion. Facilitates intercellular communication via plasmodesmata. Acts as a barrier against pathogens and prevents water loss. Stores carbohydrates for energy. Endomembrane system. The endoplasmic reticulum, ER, Golgi complex, lysosomes, and vacuoles collectively form the endomembrane system within cells. Cell structure and function. Endoplasmic reticulum, ER. The endoplasmic reticulum is a network of tubular structures dispersed throughout the cytoplasm. It exists in two forms: rough ER, RER, which has ribosomes on its surface, and smooth ER, SER, which lacks ribosomes. RER is involved in protein synthesis and secretion, while SR plays a role in lipid synthesis and detoxification. Golgi apparatus processes and packages proteins and lipids received from the ER, synthesizing glycoproteins and glycolipids. Lysosomes contain enzymes that break down macromolecules and participate in autophagy, phagocytosis, and apoptosis. Vacuoles store water, ions, nutrients, and waste, playing a role in digestion and maintaining turgor pressure in plant cells. Vesicles transport molecules between organelles and mediate cellular processes like secretion and endocytosis. Mitochondria structure. Cylindrical organelles with double membranes. The inner membrane folds into cristae, and the matrix contains dense material. Functions. aerobic respiration, site of ATP production from oxygen and nutrients. ATP production, the primary source of cellular energy. Genetic material, contains its own DNA, ribosomes, and the machinery for protein synthesis. Apoptosis, plays a role in programmed cell death, maintaining cellular homeostasis. Detoxification, converts toxic ammonia into less harmful substances in liver cells. Plastids. Plastids are double membrane bound organelles found in plant cells and euglenoids. They contain their own DNA and ribosomes and play essential roles in various cellular processes. Plastids are classified into three main types based on the pigments they contain. Chloroplasts, double membrane bound organelles that contain chlorophyll and carotenoids for capturing light energy. Responsible for photosynthesis in plant cells, consist of a stroma surrounded by thylakoids, which are organized into grana. Chromoplasts contain pigments like carotene and xanthophylls, giving plant parts yellow, orange, or red colors. Involved in the synthesis and storage of pigments that contribute to the coloration of fruits, flowers, and other plant structures. Leucoplasts, colorless plastids primarily involved in storage, can be categorized into amyloplasts. Store carbohydrates, mainly starch, such as in potatoes. Eleoplasts, store oils and fats. Aleuroplasts, store proteins, especially in seeds. Next is cytoskeleton. The cytoskeleton is a dynamic network of protein filaments within the cytoplasm of eukaryotic cells, which plays several critical roles. Microtubules provide structural support and serve as tracks for intracellular transport. Intermediate filaments maintain cell shape and integrity. Microfilaments facilitate cell movement and division. The cytoskeleton is essential for maintaining cell shape, enabling cell movement, and facilitating intracellular transport and division. Here is the diagram of cytoskeleton. See the image. Ribosomes. Ribosomes are the cellular structures where protein synthesis takes place. They are made of RNA and proteins and are either free-floating in the cytoplasm or attached to the endoplasmic reticulum. Eukaryotic ribosomes are larger, 80S, than bacterial ribosomes, 70S, and consist of two subunits: large subunit, 60S, small subunit, 40S. Ribosomes translate genetic information from mRNA into proteins, which are crucial for various cellular processes. Next is cilia and flagella. Cilia and flagella are hair-like projections that extend from the cell membrane to aid in movement. Cilia, short and numerous, they move fluid around the cell or help in locomotion. Flagella, longer and fewer in number, responsible for propelling the cell. Both structures have a 9+2 microtubule arrangement with 9 doublets surrounding a central pair. Centrosome and centrioles. The centrosome is an organelle that organizes microtubules during cell division. It contains 2 centrioles, which are cylindrical structures made of 9 triplets of microtubules arranged in a cartwheel-like pattern. Centrioles assist in forming the spindle fibers that separate chromosomes during cell division. Nucleus. The nucleus is a membrane-bound organelle found in eukaryotic cells, which houses the cell's genetic material and regulates gene expression. It plays a vital role in genetic regulation, protein synthesis, and cell division, ensuring the cell's proper functioning and integrity. Structure of the nucleus. Nuclear membrane, nuclear envelope. A double-layered membrane that surrounds the nucleus, separating its contents from the cytoplasm. The outer membrane is continuous with the endoplasmic reticulum. Nuclear pores in the membrane allow the selective exchange of materials between the nucleus and cytoplasm. Nucleoplasm. The gel-like substance within the nucleus, similar to cytoplasm. Contains water, ions, enzymes, and dissolved molecules, facilitating cellular activities within the nucleus. Chromosomes. Thread-like structures made of DNA complexed with proteins called histones. Chromosomes carry genetic information in the form of genes, which determine an organism's hereditary traits. During cell division, chromosomes condense into distinct structures visible under a microscope. Nucleolus, a dense, membraneless structure inside the nucleus, involved in synthesizing and assembling ribosomal RNA, rRNA, and ribosomal subunits, which are crucial for protein synthesis. Typically, one or more nucleoli are present in the nucleus and they contain DNA, RNA, and proteins. Thanks for watching the video. We will drop the notes in the description of this video. If you found this video helpful, please like, subscribe, and share. See you in the next video, and all the best for your exam.